Hi all, in this video we are going to discuss about the dicot root as well as the monocot root and their differences. So first we can start with the dicot root. So now the outward structure or the external surface, the external protective surface that is present in dicot root is considered as rhizodermis. So rhizodermis or epidermis, okay, understood? Now these rhizodermis, they contain finger-like projections which is called as root hair. You already know about this. This root hair is helpful in the absorption of water and mineral salts which is called as sap. Now followed by the rhizodermis or epidermis, you can see the cortex. Now what is this cortex and what kind of cells these cortex are made up of? So we can write here. So cortex. The cortex, it is made up of parenchymatous cells. Parenchymatous cells, and they also contains leucoplast. Understood? Now what is meant by this parenchymatous cells? Now in your lower classes you will be studying about two important kind of tissues. One is meristematic tissues and permanent tissues. Parenchymatous cells belongs to one of these permanent tissues. These permanent tissues are classified into three types. One is parenchyma, collenchyma and sclerenchyma. Now this cortex is made up of which type of cells? Parenchymatous cells. Now we are going to see the reason why this cortex is made up of parenchymatous cells. The next thing is leucoplast. What is leucoplast? Leuco means colorless. It is indicating colorless. Now leucoplast. So now you all know about plastids. There are three important kinds of plastids. One is chloroplast, which gives green color to all of the plant body. Chromoplast. The next one is chromoplast, which give other colors except green, other kind of colors like pink, yellow, green, pink, yellow, blue, like that to flowers and fruits. And the last one is leucoplast. This leucoplast does not have any kind of pigment, so it is colorless. At the same time, it is concerned with the storage of starch. Understood? Now even in parenchyma there will be many large vacuole will be there. So if the vacuole is so large in the cells, then it is concerned with the storage of food. So the cortex comprises many large vacuoles and it also concerned with the storage of starch or any kind of proteinaceous food. Now what are the two important things, components of cortex? That is, the cortex is made up of parenchymatous cells and it comprises leucoplast, which is concerned with the storage of food. Understood? Now where this cortex is present? It is present just near to, or it is just present below the rhizodermis or epidermis. Now rhizodermis, it is the outer protective layer of the root cell. Now in this rhizodermis only we can observe the presence of finger like root hair. Followed by rhizodermis, we can see the cortex. This cortex is made up of parenchymatous cells and leucoplast, and its major function is to store food in the form of a starch. Now comes from the cortex. So the followed by cortex, we can see the endodermis. So what is this endodermis? The inner layer of the root cell. Now this endodermis comprises two important cells, which is called Casparian strip and passage cells. What is this? Casparian strip. Casparian strip and passage cells. Now we can see about this Casparian strips. Now where it is present, students? These Casparian strip and passage cells are present on the outer layer of endodermis. Okay. Now Casparian strips, they are made up of a chemical called lignin and suberin. What is it called? Suberin. Lignin and suberin. Okay, SUBERINE, suberin. Now this Casparian strip will not allow any movement of water inside the endodermis. Okay, it will not allow water inside the endodermis. It will not allow water. Understood? They are hydrophobic in nature. Now what about this passage cells? These passage cells, as the name suggests, they easily allow the water inside the endodermis. Okay, so this is the passage cells. So they easily allow the water inside the endodermis. Now if we look closer into the endodermis, what kind of structure is present? So see here, there are certain kind of structure which is polygonal in shape. Wait a minute. So this is polygonal in shape. So now what is this polygonal shaped structure? This is xylem. Xylem. And at the same time, so only at dicot root the xylem will be in the form of a polygonal shaped structure. Understood? Now at the same time, here how many polygonal xylem cells are there? 1, 2, 3, 4. Here also 1, 2, 3, 4. Now 4 polygonal xylem cells are there. So it is considered as tetrarch. What is it called? Tetrarch. So this is called as tetrarch. Tetra means 4. Tetrarch xylem is observed. Tetrarch polygonal xylem is observed in dicot root. Understood? Now where will be the phloem will be present? So this is the phloem. So it is present near to the apex. So this is phloem. So 1, 2, 3, 4. 4 phloem cells are there. Now altogether it constitutes the vascular bundle. Understood? Now here other than the xylem and phloem, there are certain cells are present here. Okay, see here, this forms the conjunctive tissues. This forms the conjunctive tissue. Understood, students? So now we can just see the difference here. Okay, so the xylem is tetrarch in dicot root, so that is they are going to be polygonal in nature, at the same time they are tetrarch. Okay, and the pith is usually absent. Pith means the central part is usually absent. What is a pith? A pith is a central part. Okay, so even in our organs, in our human organs also, there will be a central part which is called as lumen. What is it called? Lumen. So each and every organ will be having a central part which is called as a lumen. Likewise, the each and every tissue of the plant will be having a center part that is called as a pith. Okay, here in dicot roots the pith is usually absent, and metaxylem vessels are generally polygonal in cross section. Now see here, what is the metaxylem? So this is the metaxylem. So how the shape has been, it is polygonal. So that's what I have told you, that xylem will be polygonal and tetrarch. Okay, and this is the metaxylem. Okay, so the metaxylem vessels are generally polygonal, and conjunctive tissue is usually parenchymatous in dicot roots. So see here, so the conjunctive tissue, what is the conjunctive tissue? So inside the endodermis, other than xylem and phloem, there are certain tissues are present. So these are considered as the conjunctive tissues, and they are parenchymatous. They consist parenchymatous cells. So important. And at the same time, the secondary growth is generally present. If the secondary growth is generally present, then the xylem and phloem will be have the presence of cambium. So see here, so there will be a presence of a structure called cambium. So now this part is the phloem, and now the lower part is called as the xylem. So how the xylem will be? So they will be drawing like a mesh like structure like this. So this is xylem. Now see here, in between the phloem and xylem you can observe a special kind of cells that is called as cambium. This cambium, why it is present here? Because in dicot root this cambium is essential for the production of secondary vascular bundle. That is, it is responsible and essential for production of secondary growth. So for that only this cambium is present in between phloem and xylem. Now this kind of vascular bundle is called open vascular bundle. Open vascular bundle. So just remember as OC. Open vascular bundle means what is going to there, what is going to be present there? Cambium is going to be present there. Understood? So this is the open vascular bundle system in dicot root. So xylem is usually tetrarch, that is 4 kind of xylem will be there. Pith is usually absent, the central point is usually absent. The metaxylem vessels are going to be polygonal. So why it is called as a metaxylem? See here, so this is protoxylem, this is metaxylem. Okay, and next one is conjunctive tissue is usually parenchymatous, and the secondary growth is generally present. That means the vascular bundle is going to be open vascular bundle. Okay, because of the presence of cambium. Only when there is the presence of cambium the secondary growth will be possible. Understood? So now we can discuss about the monocot roots. Okay, now we can discuss about the monocot root. So see here, the structure what we have discussed in dicot root is going to be similar as even in the structure is going to be similar in monocot root also. Okay, ரெண்டுமே ஒரே மாதிரி structure தான். So see here also there is Casparian strips is there, and then endodermis, then protoxylem, metaxylem, everything is there. But what is the difference here? Okay, see here also cortex, and there we discussed about rhizodermis or epidermis, but here in monocot root the outer protective layer is called as the epiblemma. What is it called? Epiblemma. And here also there is in at the outer epiplema region there is a finger like projection which is called as the root hair, unicellular root hair. Okay, right. So then there is followed by the cortex. So now we can just observe the difference alone. Here in dicot root I have told you that the metaxylem is going to be polygonal in structure, like this it is going to be present. Okay, but here in case of monocot root the metaxylem is going to be circular in nature. Okay, so the metaxylem is going to be circular in nature. Can you understand the difference? And at the same time the xylem cells here are present many number. For example, in dicot root I have told you the xylem is usually tetrarch, that is only 4 xylem cells are present, 4. But here see here 1 2 3 4 5 6 7 8, so more than 4 xylem cells are there. So that's why it is called as polyarch. What is it called? Polyarch xylem. Okay, understood? So we can just see the differences alone here. The in monocot roots the xylem is polyarch, that is there are many xylem cells are there. At the same time the pith is usually large at the center. See here, so this is the pith. So the pith is present here and at the same time it is so large, but in case of dicot root the pith is usually absent. Now metaxylem vessels are generally circular in section, circular, but in case of dicot root it is polygonal. Understood? Here the conjunctive tissue in monocot root is made up of scleran chimatous cells. Scleran chimatous, I have told you, no, there are three types of permanent tissue. In that the first one is parenchymatous cells, which is in dicot root the conjunctive tissue is made up of parenchyma. But here in case of monocot roots the conjunctive tissue is made up of sclerenchyma, that is the sclerenchyma cells are considered as the dead cells because of the absence of nucleus. So the conjunctive tissue is sclerenchymatous in maize solemn. Okay, and at the same time there is no secondary growth. That means what will be the vascular bundle? So just guess. So I will pause for some time. The vascular bundle is going to be closed. So the vascular bundle is going to be closed type of vascular bundle. Okay, because there won't be production of cambium. So just like this the cells are going to be arranged and at the same time the xylem and phloem will meet each other easily. So there won't be any kind of cambium will be present. Only if cambium is present secondary growth is possible, which we observed in dicot root. But here in case of monocot root the xylem there is no any kind of cambium is present, so that's why it is called as closed vascular bundle. No secondary growth is present. So this is the phloem and this is xylem. So in between there is no any kind of cambium is present. Understood? So these are the main important differences between monocot as well as dicot root. Okay, thank you.